Everyone, hello. I welcome you all to our official YouTube channel, BioCogniz. And so today we all are here to discuss about next generation sequencing and its applications, and maybe we'll also give information about some of the exciting opportunities in this field. So let's get started. So the agenda of today's webinar is, so before we get into NGS, I would like to give an introduction to BioCogniz, why we are existing and the team behind BioCogniz, and we will also share our story and a little bit about our mission and vision. So then we'll discuss about NGS, different platforms and how NGS data looks like, and we will also give an introduction to some of the applications in healthcare domains, specifically NGS in cancer research. And like I told you, we have put some exciting opportunities together for the students who are looking for roles in this NGS field. And we will also let you know the importance of acquiring NGS skills uh, in the current time. Okay. So about BioCogniz, so I am Dr. Anusha Parachuri. I'm a PhD graduate from IIT Madras, Chennai, India, and later I have moved to USA, and I'm currently a senior scientist at the Ohio State University with major focus in cancer research. So, and my friend Dakshinamurthy Sivakumar, he's a PhD in structural biology, structural bioinformatics, and he has dynamic experience in both academy and industry with great knowledge in drug discovery. So we teamed up together to build BioCogniz, and our interests are majorly next generation sequencing, AI, and computer aided drug designing. So we have two arms of BioCogniz. So one is training arm, and the other one is research R&D. So in the R&D domain, we are focusing on to provide end-to-end solutions incorporating NGS, AI, and computer-aided drug designing to handle several challenges in healthcare domain. And onto the training arm, so we have during the process of acquiring skills for our R&D team, so we have noticed a serious gap. We have noticed a serious gap where the skills are lacking. So, I mean, the serious gap between the concepts learned in the academy and the industrial application, and that's the reason behind the. I'm just looking. Sorry, there is some technical issue. I'm trying to share my screen once again. So my voice is audible. Like, you can hear my voice. So, yeah, this is about BioCogniz, the training arm and the research arm, and the major focuses are NGS, AI, and computer-aided drug design. So let's get started with next generation sequencing. So for someone who are completely new to this topic, next generation sequencing, so in simplest terms, so NGS is a technology which is applied to understand the composition of our genetic material present in our cells. So genetic material could be in different forms like DNA, RNA. So understanding any of these genetic materials in our cells, the technique applied is next generation sequencing. So let's briefly go into the history of our DNA. So where it all started. So around in 1950s, the first time the Watson and Crick discovered the three-dimensional figure of genetic material, that is DNA, the double helix structure, and we got to understand it in a much better way by applying it in the research. And so that is where it all started. So discovering the three-dimensional structure of DNA in 1950s, and hence from then a lot of advancements happened. And around in 1970s, Frederick Sanger, he first sequenced a short sequence of DNA to understand the composition of it and the role of genetic material in our cells. And so that is all. That is where around in 1970s the sequencing technique started, and hence evolution of these techniques advanced. And the current technique is next generation sequencing. So maybe I'll go in detail about these in the coming slides. But yeah, in the current next generation sequencing techniques, we can simultaneously sequence millions and billions of DNA fragments simultaneously, generating lots of data to understand how our cells function or how everything else function. For example, so why we all look so different, or why our hair color is gray or black or any other color, or why our eye color is different among each of us. So, to answer all of these questions and similar other questions in healthcare, so we apply NGS techniques. So let's look at the evolution of NGS techniques, like how, like when it started, and what's the current situation of NGS techniques. So, like I said earlier, around in 1970s, the first scientist Frederick Sanger sequenced a short length of, a short DNA fragment in the bacteria from the bacterial genome, which is a lac operator. So it all started then. So the scientists applied a technique called Sanger sequencing, so where we sequence one base by base, and the first sequence which was published using this Sanger sequencing was 24 nucleotide long. So that is where it started, and then the process, so the process was like base by base. So then the process was automated in the later stages, and it evolved, and the next technique was maximum Gilbert. It is a little advanced technique of Sanger sequencing. And then in 19 around the 1990s, so there is this international effort of understanding our human genome. So what exactly our human cell, the genetic material of our human cells looks like. So that was a question scientists across the globe asked, and it was an international collaboration. And the proposal was to sequence the entire human genome. So the Human Genome Project was started in around like 1990, and the technique implemented in the project was Sanger sequencing. So it took like around like 13 long years to sequence one human genome. I mean, just one human genome, and and now, at this current time, by applying the current technology of NGS techniques, we can now sequence one human genome in maybe a matter of hours or a few days to just sequence one human genome. So that is how we have advanced in sequencing techniques, and all the techniques in between after Human Genome Project was like pyro sequencing. It's a different technique, but little advanced than the Sanger sequencing. And then the current technique, which is widely used in the industry or in the academy, is Illumina sequencing. That is a technique. The sequencing is done by sequencing by synthesis, technique, and the major player in the market is Illumina. So 80% of the sequencing market is managed by Illumina providers, and rest of the providers are Ion Torrent, and each of the platform comes with a different technology and different advancements. But the most commonly used one is Illumina sequencing. And at the current times, it's Oxford Nanopore and PacBio. There's a little advancements on that end. By so Illumina platform, we can sequence so a stretch of 100 to 300 base pairs. Whereas in the latest technology from Oxford or PacBio, we can sequence a long stretch of DNA fragments ranging from somewhere between 1000 to 20,000 bases. So that is the advancement in the current time, but still Illumina is a major player in the market, a sequence like short read sequencing of around 100 to 300 base pairs. So let's look at some of the Illumina, what are like different levels of sequencing techniques. So maybe all the Sanger sequencing, the Maxim Gilbert, and all the automation of Sanger sequencing, if you all put it together, so we call it like first generation sequencing techniques. It was all started there, and then the next generation sequencing came into play. So where Illumina is a major provider, and the others are like 454, Pyro sequencing, Ion Torrent, and Solexa platforms. So where the maximum we can sequence is up to 500 base pairs long of DNA fragments, and we can automate it at different levels. So simultaneously, millions and billions of DNA fragments can be sequenced using these techniques, which are called next generation sequencing techniques. And now the third generation sequencing, so all the long read sequencing coming from PacBio and Oxford Nanopore sequencing techniques. We call it like third generation sequencing. So there is a lot of refinement has to be done, because when we compare the second generation sequencing techniques with the third generation, the latest techniques, so there is a lot of error, the quality issues. So Illumina is known to provide the highest quality of sequencing data compared to PacBio and Oxford long read sequencing, where the quality is a little bit in the lower end. So now, since Illumina is the commonly used platform, let's look different versions of platforms available from Illumina. So Illumina has different versions of sequencing platforms, starting with the MiniSeq or MySeq series. The MiniSeq and the MySeq series are the smallest versions of the or the portable versions of the sequencing platforms from Illumina, where which can be used regularly in our daily research. So at a time we could sequence one or two genomes or exomes in one run. Whereas the advanced versions are NextSeq, HiSeq, and the NovaSeq, where multiple samples can be sequenced in single run. And the NovaSeq is the latest version from Illumina, where we can do population based, and I think NovaSeq can generate around like 20,000 genome sequences in a year. So that is the advancement happened in the Illumina platforms and the settings of sequencing genetic material. So now I'm talking about the sequencing and different platforms. So now let's see the principle behind NGS, like how it works. So exactly. So the now I'm sure you all understand like NGS is to know the sequence composition of our DNA or our genetic material in our cells. So how exactly are we going to do it? Right, so the sample, we can extract the genetic material from variety of samples. So for example, the sample could be a blood sample or a tissue sample or a saliva sample, if we are trying to understand the genetic material of human samples. And if we want to understand the genetic material of microbes present in our environment, so for example, the microbes in the soil, so the soil could be a sample. And like that, like there could be many types of samples from which we can extract the genetic material. So nucleic the first step is extracting the genetic material, let it be DNA or RNA. So then the quality check on the wet lab side. So then the second major step of NGS workflow is library preparation. So when we extract the DNA or the genetic material, we have amount of DNA present in our hands. So then the technique works like fragmenting the entire DNA using certain techniques on the wet lab side, and we add adapters and indexes accordingly according to the problem we are trying to solve, and we amplify it for improving our confidence in sequencing. So once that step is done, the third step is sequencing. So after all the DNA or the genetic material is processed, we load the sample onto our flow cell, or that's a equipment on the sequencing machine. So we load the samples onto that, and that flow cell is inserted into the sequencing machine in order to get the information of what bases are present in the DNA sample we are analyzing. So all the sequences or all the bases are read based on fluorescence technique, and specifically on Illumina, it is sequencing by synthesis. So when each base gets added to the flow cell or the primers, so then it's get recorded, and that is how we get the raw data files. So the raw data files contain all the information of what the composition of our DNA fragments present in our sample. So all that is given as a raw sequencing file. Then the last step is a data analysis. So now we have the raw data files. So as a computational biologist, like our role starts here, begins here, where we start working with the raw data files, quickly do the quality check and aligning it with the reference genome to check where the genetic changes are present in the sample of our interest, and the further downstream analysis. So majorly it is all four steps: DNA or nucleic acid extraction, then library preparation, then sequencing, and data analysis. So some of the sequencing techniques are the ones which I listed here. There are So some of the sequencing techniques are here: whole genome, whole exome. So basically, if you classify the genetic material broadly, one is DNA and the other is RNA. So understanding the DNA, we have various techniques. So we choose the technique based on the research question we are trying to address. So for DNA, the commonly used techniques are whole genome sequencing, whole exome sequencing, targeted sequencing. So all these genome, exome, and targeted sequencing is to understand certain parts of our genetic or DNA material. So when I say whole genome sequencing, it is trying to understand the composition of our entire genetic material present in our cells. Whereas whole exome sequencing is just to understand the coding part of our DNA, and targeted sequencing is if I say, like, our entire genetic material is made up of around, I would say, like 23,000 to 25,000 genes. So if our interest is a set of 10 genes, then we go with targeted sequencing. So that is how we pick the sequencing technique according to our interest. And then the other techniques for DNA study is ChIP-seq, ATAC-seq, and bisulfite sequencing. So those are majorly used in epigenomics, where we try to understand the methylation patterns of DNA. So there are these kind of methylation patterns which regulate downstream processes in our cells. So it is these techniques are used to understand the methylation patterns of DNA. And then the RNA sequencing is called transcriptome sequencing. So when if we want to understand the composition of our entire RNA, we call it whole transcriptome sequencing. And if it is only a specific region of RNA, we call it targeted RNA sequencing. And then microbiome sequencing or metagenomics. where we try to understand the genomic sequences of bacteria, fungi, viruses, and all kinds of microbiomes. We call it metagenomic sequencing, and the sequencing techniques are 16S rRNA and 18S RNA sequencing. And then there is this advanced sequencing technique called single cell sequencing, where we sequence the genetic material at at a single cell resolution. So this is just to give an overview of what are all the sequencing techniques available uh, for understanding the DNA or RNA in our cells. So now there are so many techniques. So what kind of information or data can we expect from these techniques? So I mean, like we apply each of these techniques for a purpose to solve certain kind of research questions, and there is certain kind of information coming out of all of these analysis techniques or sequencing techniques. So, for example, DNA whole exome or whole genome sequencing, majorly we look at the genetic changes present in the sample of our interest. So, we compare the sample of our interest with the reference genome, so which is generated in our human in our case it is the reference genome came out of human genome project, where now we have the composition of our human genome and the composition of human genome present in the normal individuals. So now when I'm analyzing a sample of a disordered sample. So then I compare the genome of that with the reference genome, and then the prominent ones which we look at in the whole genome or exome sequencing is point mutations. So these are different kinds of mutations which are listed here, like point mutations, indels, copy number changes. So it is like wherever the genetic changes are seen are all reported from whole genome or whole exome sequencing. And transcriptome sequencing is like sequencing the RNA, where this is done at a certain conditions. If you want to compare a different treatment condition, say how the treatment work. I mean, like how the genes are expressing before the treatment and after the treatment. Is there any kind of improvement, or what are the set of genes which are involved in a disorder, or those kind of information we apply transcriptomics or RNA sequencing. And then, like I already mentioned, epigenomics is like to understand the methylation patterns, and to also understand the protein DNA interactions. We use this ChIP-seq, and ATAC-seq is for understanding the regulatory regions of our DNA. So, and then the latest hot topic in NGS is the metagenomics, where we try to understand the composition of microbiomes. So, given a soil sample or given a gut microbiome sample, like where we have a diverse set of microbiomes present in those samples, right? So, to understand that, and the other another important or interesting application is this antibiotic resistance. So these days we hear a lot about this antibiotic resistance, right? So what are the bacteria prone to this resistance? And to understand these kind of problems, we apply metagenomics, and we get the information of the different microbiome profiles from the sequencing techniques. So then the advanced level of analysis is integrating all of these. So when if I have a sample, like different types of samples of same disorder, so then I would like to get a collective information or comprehensive picture of that disorder by integrating the analysis from genomics, transcriptomics, and so on. Then here comes a question. So now I know what is NGS, and there are different platforms for NGS. So now my question would be like, how exactly NGS data looks like? I know, like I do the sequencing and we get the raw files. So what exactly is present in the raw files? So these days, like most of the technologies, the raw file we get out of NGS data is a FASTQ file. So FASTQ is a kind of data format. It's a text-based format where all the sequencing information of all the DNA we are analyzing on the sequencing machine is stored in a text file in FASTQ format. So the one which I'm representing here on the screen is a FASTQ format of a sequence. So if you see, there is a, I mean, there is a meaning for this format, and it is encrypted in this or encoded in this format to make it easy for computational biologists to work on. And so each line within that FASTQ file has a meaning and a purpose for it. So the first line, so every set of 4 lines represents one DNA fragment of our sample. Like I mentioned in the earlier workflow, so the second step is library preparation where we fragment our DNA, right? So in our FASTQ file, we have all those fragments represented in a set of 4 lines. And here we call it as a read. So a read is nothing but a DNA fragment on the wet lab site. So now, for every set of 4 lines, the first line is all about the sequencing machine and all about the length of the sequencing read. And then the second line is your actual sequence of the fragment. And the third line is just a plus symbol. Sometimes it has the information repeated what's present on this line 1. Otherwise, it's plus and empty. And the fourth line is all about the quality scores, which is really important information for us. So the fourth line and each alphabet has a meaning for it. So each alphabet represents a quality score for each base on this fragment, right? So like this, all these first 4 lines is for one fragment and the next 4 lines is for the other fragment. So just imagine, so if I have billions of reads from my sample of analysis, so just imagine how much amount of data am I going to generate from NGS. So it is like if we are sequencing one whole genome of our cells, then we are going to generate on an average, with an average quality, we are going to generate around like 200 to 240 GB of data for just one sample. And again, if it is whole exome sequencing, so a whole exome sequencing is just sequencing the coding part of DNA. So I guess you all know, like coding part of DNA is just 2% of our entire DNA. So when we just sequence that 1 to 2% of the entire DNA, that is whole exome sequencing, on an average quality, we will be generating around like 20 to 40 GB of data. So now the technology is so advanced that I can simultaneously sequence multiple samples. So if I'm sequencing like 10 or 20 samples in one run, just imagine how much amount of data we are generating for whole exome sequencing or whole genome sequencing, and how effective that would be. So now so much of data and so much of complexity, and so many fragments to be analyzed. So now the next question would be like how to analyze this complex NGS data. So now, no worries. For us, computational biologists, we have plenty of computational tools available in the literature, and which are open for us for public use, where we can apply different techniques to analyze this complex NGS data. And for each application, there are like specific tools. If you're working with whole exome or whole genome sequencing, you have a set of tools for it. And if you're working with metagenomic samples, we have a set of tools for that as well. So we have no worries. So just that we have to acquire the skills to learn the tools or techniques to analyze this complex data to infer some meaningful observations from the data we work with. So this picture which I am representing here on the screen shows different levels of computational analysis of NGS data. So the level 1 or stage 1 is this. So where we get the raw sequence data or the raw calls. So where we check the quality of those base calls and do the refinement and all of that, and then we get the refined FASTQ files from this primary analysis, or you call it stage 1 analysis. So once you get the refined reads or the refined fragments in your FASTQ file, so then the next level of analysis is your stage 2 analysis or secondary analysis, where we try to align, because we just have the reads and fragments in our sample, right? So we don't know from where exactly these reads are coming from. So in our cells, we have around like uh, chromosomes, like 22 chromosomes. So we don't know from which chromosome exactly that read is coming from and which gene it is coming from. So there are genes which are placed on these chromosomes, and we have two copies of each gene in our cells. So to get all of that information, this secondary analysis has to be done. So where we align all the raw DNA fragments to our reference genome, then check where exactly they are matching. So there are different computational tools for accomplishing those tasks. So some of them which are represented here, otherwise there are many in the literature. You can choose according to your research question. So once the alignment is done, now we have the alignment file with all the information of from which chromosome the read is coming, and then we refine it, removing the duplicates and all of that. And now we have a final alignment file from which we can apply again variant calling techniques, from which we can see the genetic changes. So all the raw variants until starting from the refined reads to your raw variant calls, by applying different computational techniques, we call it all secondary level analysis of NGS data. And then comes the third level of analysis, is stage 3 or tertiary level analysis, which is advanced level analysis of NGS data, where we start looking at the raw variant calls and try to refine them, just to look at which ones are real and which ones could be random, and there are techniques for it. And again, from the observed variants, which ones are really important or which ones are already known to have their role in certain disorders or something, which ones are novel, if novel, like what could be its role in that disorder. So all those kind of functional level analysis and all the functional annotations and linking them with the known information and coming with new observations is all the tertiary analysis in NGS data analysis. So to sum it up, so it is like 3 levels of analysis. Each level has its own significance, and at each level, the quality check is really important while we are working with the data to get reliable inferences from the data we look at. And the one which I mentioned here on the screen is CATK Toolkit. It's an excellent package with plenty of tools related to NGS data analysis. And you can just go to that page and look at different tools, and you would find a treasure of NGS data tools there. So to just give an example of how exactly a computational workflow looks like for whole exome sequences. So if I have a set of whole exome sequences and I have the raw data in a FASTQ format, so how exactly am I going to deal with it? So there are like certain tools. There are many tools. So the tools which I just mentioned here on the screen are the ones which are familiar to me. So the raw file is FastQ file, and we have it. I mean, we have a certain tool called FastQC. It's a quality control tool, and it's a very commonly used tool for quality check to quickly see how the sequencing quality turned out and what's the composition of GC and many other factors. So it gives a nice summary and also nice figures of the quality of sequencing. And then the trigonometric is kind of refinement, like trying to remove all the additional sequences which we have added during sample preparation on the wet lab side for different purposes. So where there are multiple tools like trigonometric and cut adapt. So where we trim these adapters, and then the next significant step here in computational analysis is the alignment. So we have to align our raw reads with the reference genome. And the tool, the commonly used tool is BWMM. It's a Barrow-Wheeler's alignment algorithm where it aligns the raw reads to the reference genome and says exactly from which position of our genome these reads are coming from. And then Sun Tools, it's a package where we can refine this alignment and again check the quality, the sequencing depth, sequencing coverage. I'm not going into details of all of those terminologies, but it has like plenty of functionalities. Sun Tools has plenty of tools within it. And then there are tools to remove the PCR duplicates, because PCR is a step on the wet lab side where we amplify for the confidence of our sequencing, and then we have to remove those duplicates to get the unique reads, and there are tools for it. And the one which I use for whole exome sequencing is RN2. And then now we have the final alignment file with the unique reads within it, and then we apply various computational tools, and the common ones are VASCAN, NewTek2 from GATK, and many others, where we can call different types of mutations. I think I guess like each of these tools are meant for different purposes. So VASCAN can give us information about single nucleotide changes. In other words, it's point mutations, and it also gives us information on copy number variations and small indels and insertions and deletions. And whereas BTEC2 also gives some reliable information of point mutations and indels. And there are other tools like Falcon, from which we get this amplification information and copy number changes, whether a particular gene is present in two copies or more than two copies or less than two copies, or what part of gene is altered, and all that kind of information we can get from applying different computational tools. So this is just to give an overview of how a computational setup looks like if we are working with whole exome sequencing samples. So likewise, we can create a workflow by applying multiple computational techniques if we are dealing with whole genome sequences, or if we are dealing with metagenomic sequences to understand the microbial communities. So there are a set of tools where we can manually put all of them together, build our own pipelines to make it more flexible rather than using something which is already existing. So as a beginning point, I would suggest like someone who are interested to step into NGS data analysis, or who are interested to gain these skills, I would recommend to start quickly with some easy to use tools like cloud-based web servers. I think one of the popular cloud-based web server is a Galaxy website. So where it's a valuable resource for a computational biologist who is working with NGS data. It has a lot of tools within it. So as a first step, I would recommend anyone who are interested to work with this to get hands on the Galaxy server, and there are also very nice tutorials present on the Galaxy community page, where it's clearly shown how the certain type of NGS data can be analyzed on that platform. So now that we know what NGS is and how exactly NGS data, raw data looks like, and what are different computational approaches to tackle NGS data. Now, the next question is like, what is the application? Maybe to get an to give an overview of different applications of NGS, we will start with applications of NGS in healthcare domain. So how and why exactly NGS is applied? I know NGS is understanding the DNA composition or RNA composition. Okay, now I know the composition of RNA. Now I know the variants. So how exactly am I going to implement it in the real time? Like what exactly is the application of understanding all of these variants, right? So for example, the ones which I listed here are some of the important applications of NGS in healthcare domain. So the first one is diagnosis. So in diagnosis, where we try to understand the genetic basis of the disease. So what exactly is the genetic composition of the genes or the sample of a disordered individual versus the normal, right? So then we can identify certain biomarkers which could be the reason behind the risk. So in that way, we can also do the risk assessment for several disorders. And the other application is clinical monitoring. So we can also answer this question like how a patient is responding to a particular treatment. So how exactly the genes or the composition of genes are changing before the treatment versus after the treatment. So we can do that clinical monitoring, but majorly in the real time and in the clinical settings. This is being applied in cancer clinical monitoring in different locations across the globe. Otherwise, in other disorders, it is coming up, but not yet applied in the real time in the clinical settings. And then the next interesting application is personalized medicine or precision medicine. So when a patient doesn't respond to any kind of treatment, or when we want to understand why a patient is not responding to a certain treatment. So to answer all of these questions, again, we can apply NGS. So there could be some kind of biomarkers that are playing an important role, which are affecting this response to the treatment for certain patients versus the other group of patients. To answer all of these, so these are where some of the situations where we apply NGS techniques to all of these healthcare problems. So let's look at one of the prominent example where NGS is applied, and in fact, NGS is started being applied in healthcare domain with cancer. So I mean, like it's very popular of its application in cancer research. So why exactly it is? Because the cancer is not prone to a lot of mutations. So usually we see lot of mutations in cancer patients, and that is why we apply NGS to understand the dynamics of mutations and the composition of mutations in different genes. And over the past decade of our research in cancer applying NGS, so this is the advancement which I'm showing here is a nice figure from a recent research publication where nice representation of different genes which we have identified over a period of 10 to 15 years now. So if you see each of the cancer type, now we have a set of genes which are known to be uh, some way linked with the progression of the disease. So for blood cancer, these are some of the important genes which are known, and these are some of the known genes or biomarkers. In other words, for lung cancer and for the liver cancer, these are some of the important genes which are playing a role in cancer progression and many other things. And likewise, so this is like wealth of information on the genes and the biomarkers which are involved in different cancer types, and this was all possible, and all the data is available because of advancements in next generation sequencing techniques. And since we are talking about cancer research, and my major focus is cancer research, and I'm actively involved in cancer research, like my one of the major questions which we are trying to ask with the data we have is, so most of the times the cancer is detected in the later stages, let's say stage 3 and stage 4, so where the cancer is already progressed to multiple organ sites. So the question we are trying to address is how exactly this is happening. So, how exactly the metastasis is happening, and what are the root causes, or what are the mechanisms behind this metastasis or the progression of cancer? So, to address this problem, we are utilizing rapid research autopsy as a powerful tool. So, at the Ohio State University, where I'm actively involved in cancer research, so we have a rapid research autopsy protocol where we get consent from the patients, and once a patient passed away from the cancer, we get the human body transported to our autopsy center where we do perform rapid research autopsy, and we collect samples from multiple locations. So if you look at the image or the picture here on your right side of your screen, so just compare the clinical biopsy versus the research autopsy. So I mean, I would like to highlight the importance of research autopsy here. So when we collect the samples from biopsy, so the sample is very limited, so we only get a certain part of the entire cancer tissue. So there we are capturing only a certain percentage of cancer, right? So we're not, so we are missing out many of the cells from the clinical biopsy. Whereas in the research autopsy, we have wealth of information or wealth of samples. We can collect samples from multiple locations, and that helps us to get a comprehensive picture of cancer, how cancer initiated, how the cancer looks like in the primary site versus how the cancer looks like in other progress sites. So, understanding all of these cells and the genetic materials, we apply NGS techniques to understand the composition of genetic material present in the primary site versus all the other metastatic sites to come to get a clear picture of how the genes and what's the diversity between the genes in different organ sites. So this gives us a comprehensive understanding of metastatic cancer from the patients. So one of my published research works on lung cancer research. So this is where we have applied rapid research autopsy, where we collected samples from 5 different patients who passed away with small cell lung cancer, and we collected samples from different organ sites. to try to understand the metastatic sites. And interestingly, what we observed is there is a lot of tumor heterogeneity present across different sites of cancer. So that means that there are different types of cells within a single tumor tissue. So likewise, if you look at just one patient data on this figure, on this picture here, if you look at just A, patient A, so these are all the samples which we collected during research autopsy. And what we observed is there are like at least six different groups of cells carrying unique set of mutations. So that means the point here, what the interesting point here is, there is not a common set of mutations present across all the tumors. Rather, there are different set of mutations present at different sites or different metastatic sites. So that answers all. Like, I mean, like, this is one of the serious problem in cancer, which causes which leads to this heterogeneity, and some of the cells respond. So that is why we hear that some of the cells respond to the treatment and some of them not, because we are only targeting the cells with a certain mutation and not all the cells, right? Because the mutations are varying across the cells. So likewise, interestingly, for all the five patients, we observed significant tumor heterogeneity and also the evolution of genetic mutations. So we applied certain computational techniques to understand how the genetic mutations are evolving or are evolved over the period of time. So we also have the primary biopsy samples of these patients during the treatment different time points. So we compare the primary sample from the diagnosis versus the samples during the treatment versus the samples from the autopsy to get a comprehensive picture of how the cancer is evolving or how the gene mutations are evolving or getting acquired over the period of time as the cancer is progressing. So this is an interesting study to get a nice understanding of how metastasis mechanisms within lung cancer. Likewise, we are also working on different cancer types like gastrointestinal stromal tumor, liver cancer, and so on. So this is just to give an understanding or an introduction of how NGS is being applied. So we have applied NGS technique, different NGS techniques like whole genome sequencing, whole exome sequencing of all of these samples, and we also performed RNA sequencing of some of these samples to understand the dynamics of gene expressions compared to the normal. So it's like we kind of integrated multi-omics platforms like integrated the DNA and RNA analysis, and we also try to understand the immune environment of these patients and so on. So this is how, I mean, this is how NGS can help us answer a lot of questions at different levels of our analysis to get interesting insights of certain disorders. And likewise, like cancer, there are also many other disorders in which NGS is being applied. And these are some of the disorders which I just listed here on this picture are the ones which are also applied in the real time clinical settings. So some of them are like understanding cardiovascular diseases, understanding the pharmacogenomics. So for example, what are the genes which are sensitive to certain treatment versus what are the genes resistant to certain treatment, and so on, and antimicrobial resistance. And also, NGS is applied to understand certain issues in infertility research and also different cancer types and so on. So these are some of them, and there are plenty of applications of NGS which are ongoing in the real-time setting in different domains. So I'm talking only about the healthcare domain, but there are different applications of NGS apart from healthcare, like agriculture, food industry. So where, for example, in agriculture, what is the composition of the soil where we grow the crops and how to improve it by understanding the composition of the microbiome present in the soil, or understanding the genes of the crops and all these genetically modified foods and improving the quality. So in all of these domains, NGS is helping us to understand and improve the quality of research and also in the real time. So then now we know what is NGS, we know the data and the analysis workflow and applications. So since the data is very complex, there are also a lot of challenges involved in NGS data analysis. I think the first main important challenge is this ethical considerations, because the data is very sensitive. We have to be really responsible and careful while we are analyzing the sensitive information, that is genetic information of human samples or any other samples. Since the data is very huge, plenty of data is generated from sequencing, so there are difficulties with the storage techniques, the space and all of that. So now we have a lot of cloud platforms where the data is being stored. I think one of the important or the one of the common resource of NGS is NCBI SRA, sequence read archive. So where it's a cloud-based server where a lot of NGS data is being deposited, and a lot of data is also publicly available. So I would also suggest, if someone is interested to get a hands-on, or someone is really curious to know how the data is, you can just go browse NCBI SRA database and just look at any of the disorder. Just type in there and you'll see plenty of samples and different platforms on which the samples are analyzed and all of that. So just to give an overview, and then the data and complexity. So since it's very huge and it's a lot of millions and billions of reads are generated, so and also when we are generating lots of data, I think the quality is also an issue, right? So we have to be very careful in monitoring the quality of our analysis at different levels. So these are some of the challenges while handling NGS data. And I would also like to bring up this growth in NGS market. So I would say like NGS or next generation sequencing, the real time market is experiencing rapid growth, definitely. So this is one of the latest market research report from a popular company related to NGS. So if you see, there are different applications of NGS in the market, like drug discovery, personalized medicine, like I discussed, and diagnosis of several diseases. Like for example, rare diseases, where like if you see. Or if you go through the literature related to rare diseases, where the standard techniques have failed plenty of times to diagnose what kind of disorder it is. But then NGOs helped us understand what kind of disorder it is and from where the disorder is originating and what are the important genes being involved in that disorder. So all of those applications are the real-time applications of NGOs in diagnosis and infectious diseases. I think the famous, I mean, you all are familiar with the latest pandemic, right? The coronavirus. So NGS helped us quickly to understand the genetic composition of the viral genome, the viral RNA, and then all the vaccines came up really quick into the market. Is all because I mean, like one of the important role is of NGOs there to address coronavirus problem, the pandemic, and we really came out of it quickly. And then the other applications are the agriculture research, animal research, and the environmental research, and also climatic conditions, many other applications. So these are the top applications in the real time. And by technology wise, if you see, there are like different techniques which I have just mentioned, the whole exome genome, RNA sequencing, and epigenomics. All of these technologies are the popular ones which are being applied. And if you see, and the important point we want to bring up is, so now in the next 8 years, we are going to experience 5x to 8x of fivefold or eightfold of exponential growth in NGS market, because now we started applying NGS to understand various problems in the real time. So and also the advancements in NGS technique is going to lead this path of exponential growth of NGS. So now the question is, are we really ready to handle or to fit into this growth? So we have to answer that by ourselves, if we have the skills to be fit into this market or not. So then we wanted to bring this up to all the young students here who are listening to this talk. So these are some of the NGS related roles which are put up open in different platforms like job platforms like Nature, LinkedIn, and many others. So where we see plenty of roles which are open to the students who are graduating, right? So these are the jobs which we have put up or which we have noticed in the last couple of days. So just imagine, and we also have noticed there are like jobs which are open from several months. So that means there are like jobs available, but the companies are not able to match the skill to the job. So that means it's not that we don't have graduates or from the life sciences, but that means there is no appropriate skill that's matching to the jobs, right? So this is the serious gap which we have noticed between the academic qualification versus the industrial application. I mean, the skills required for industrial application. So that is, and this is also our personal experience. So when so we are 7 months old or 8 months old now, and when we initially planned to build our R&D team at BioCogniz, we were looking at NGS skills and drug discovery skills and AI skills to get into our team. And then this is our own experience of not finding an appropriate skill to the roles present in our company, BioCogniz. And that is when we have decided to come up with this training arm of BioCogniz, where we want to address or fill in some of the gap between the academy and industry. So what we would highlight here is not just the qualification, but acquiring the skill and the quality of the skill is really important to get to be fit into the market and to match the requirement in the market. And in order to grow along with the market, we have to acquire the skills if we are from this background. And also, if so, since we are talking about the training and the importance of acquiring the NGS skills, so at BioCogniz we have many mentorship programs. So we believe each individual have a different way of learning. So some of them would be interested in learning by listening. Some of them would be interested by learning by getting hands-on training, and some of them would be interested to learn by understanding the challenge or understanding the problem and starting from there. So then we have these diverse programs of training in these three fields: NGS, next generation sequencing, AI, artificial intelligence and machine learning, and drug discovery. So we also have programs where we interlink AI and NGS, AI and drug discovery. So these are some of them. So where we have this project training and project internships for the students from diverse backgrounds, majorly from life sciences. And also we conduct regular courses on some important and potential topics of NGS and drug discovery and AI and ML. And we also have regular workshops and hands-on training. And we believe in this learning by doing approach, where we introduce the problem to the students if they are working on the projects. So we make them work on the projects while introducing the tools and techniques appropriate to handle the challenge in the projects. So we also have many group discussions and presentations where we have these regular interactions and one-on-one mentorship directly with us, with me, if it is related to NGS and AI, and Dr. Dakshinamurthy Sivakumar if it is drug discovery and AI. So we are very active on LinkedIn. So you guys, if you are interested, you can just look at our LinkedIn page, and we have around like 10,000 followers on our page. And if you are really interested in learning more about this, acquiring these NGO skills, or if you really quickly want to talk to us about how to start with or where to start with, or if you need any kind of guidance related to any of these topics, just reach us here at info@biocogniz.com and take a look at our webpage. And yeah, here we go. I'll end the session here, and we are open to take up any questions you may have. So I guess there is one question. I have one doubt. Like, is there any possibility that during trimming of adapters, can our coding part also gets removed? So there are chances because see, computational algorithms work on different different ways. So it all like I mean, but again, we can make sure to improve the quality of adapter trimming, and we can also make sure that the actual reads are not getting trimmed by tuning the parameters of the algorithms. So it is all again for your data. We have to look at so what exactly the platform used. So there are many factors involved within it. So what platform used and what kind of data it is and what are the tools you're using, and fine tuning the parameters, we can definitely achieve it. But I would not say that we can achieve 100%, but we can achieve maximum of make sure that not our the real fragments are getting out while we are doing the streaming. So any other questions, or like to know any kind of information related to our training programs, you all are welcome. So maybe we'll be open for another couple of minutes and we'll end this session. In case if you are ending the session and you still have the doubts, you can just write to us at info@biocogniz.com, and we're happy to answer any of your questions related to these topics. I hope you all understood like what we have just discussed, and I hope this is helpful to get an understanding of what NGS is for the beginners. And yeah, like I told you, like if you are going to start working with NGS data, then I would just recommend, or I believe that this works. Like if you directly go look at the data, how the data looks like in the NCBI SRA, and then get hands-on on the cloud-based platforms would give a nice experience of how you can deal with the data, and then you can start growing from there. And also on our LinkedIn page, we'll be regularly posting about the computational tools related to NGS, drug discovery, and AI. So where we put it open to all our followers on the page, so you can also get that information about the latest technology or the tools related to any of these topics. Thank you. Maybe we'll end the session, and we are open to questions on the email. Thank you.